My name is Yuvha Karthikeyan, and I'm a junior at Solon High School in Ohio. I'm so excited to be presenting my research here at the Johns Hopkins Global Health Leadership Conference. My project is titled Anatomical Characterization of the Sympathetic Trunk for Targeted Neuromodulation. So, a little bit about me. I currently work as a neuroscience research intern at Case Western Reserve University, and beyond the lab, I'm actively involved in my school's Science Olympiad, Future Problem Solvers, and HOSA clubs, where I've competed at regional, state, national, and international levels. I'm also the vice president of Women in STEM and the co-director of the Yale Gray Matter project at my school. I've also been training in Indian classical song, dance, and violin for over 10 years, and I'm also humbled and honored to have performed at prestigious venues such as Carnegie Hall and Playhouse Square. Looking ahead, I want to pursue neuroscience to help save lives, and I see this project as my first step. Neurological disorders are a leading cause of disability worldwide, impacting over 3 billion people. Conditions like chronic pain, epilepsy, Parkinson's, and depression are global health challenges. One promising approach is neuromodulation, a powerful therapeutic approach that uses electrical or chemical stimulation to regulate activity within the nervous system. However, neuromodulation doesn't always work the same way for everyone. For some patients, it brings immense relief. For others, it fails to deliver significant benefits. One key reason for this inconsistency may be the lack of personalization. Most neuromodulation systems are designed using generalized models of nerve anatomy, but in reality, the human nervous system is incredibly complex and variable. Even nerves that look similar on the outside can be structured very differently on the inside. Understanding those micro-level differences is crucial if we want to make neuromodulation therapies more precise, effective, and tailored to individual patients. This realization formed the basis of my research. I set out to study the human sympathetic nerve, a key part of the autonomic nervous system responsible for our fight or flight response. Because this trunk plays a vital role in homeostasis, understanding its fascicular microstructure could significantly improve 3D computational models used in neuromodulation therapies and surgical planning. These models often assumed uniform fascicle properties, which may not be true, and inaccuracies could compromise treatment outcomes. This research addresses this by analyzing multiple human sympathetic trunk samples to identify fascicular variations in both structure and distribution. In other words, how the small bundles of nerve fibers within the sympathetic nerve differ in shape, size, and organization between individuals and even across different parts of the body. To carry out this research, I used cadaveric human sympathetic nerve tissues obtained through the National Institute of Health's Reconstruct Vagal Anatomy Initiative. The samples were then fixed in formalin and embedded in paraffin in order to preserve the tissue and allow it to be cut into very thin slices using a microtome. These slices, each cut 4 micrometers thick, were carefully mounted onto glass slides and stained using hematoxylin and eosin to view general structure. I also used immunohistochemistry to label specific proteins that are important for identifying the makeup of the nerve. For example, I stained for myelin basic protein, which highlights the presence of myelinated fibers, and neurofilament proteins, which give structural support to neurons. I also used tyrosine hydroxylase staining to confirm the tissue's identity as part of the sympathetic nervous system. Once stained, I analyzed the slides using Napari, a Python-based library and an advanced imaging software that allowed me to measure and compare different structural features. One of the most striking images we analyzed was the immunohistochemical overlay showing both tyrosine hydroxylase, or TH, and myelin basic protein, or MBP, staining. In these images, you'll notice that the same fascicle tested positive for both TH and MBP, meaning it was both catecholinergic and heavily myelinated. Next, we observed considerable variation in fiber density across the same fascicle. Some areas had tightly packed axons, indicating the potential for fast and frequent signal transmission. Others had more loosely spaced, sparsely myelinated fibers. These regional differences suggest that even within the same fascicle, different regions may serve unique physiological roles, some optimized for seed, other optimized for modulation, regulation, or local feedback. Perhaps one of the most important findings was the distinct structural asymmetry between the left and right sympathetic trunks. The left trunk, which is positioned adjacent to the aorta, had fascicles that were generally more uniformly organized, denser, and more heavily myelinated. In contrast, the right trunk, located near the inferior vena cava, showed greater structural heterogeneity. It had fascicles that varied more dramatically in fiber composition and organization. This anatomical asymmetry may reflect functional differences between the left and right branches of the sympathetic nervous system, which has significant implications when designing site-specific stimulation strategies for patients. We also measured differences in tissue structure between individuals using segmented mass generated by Napari from multiple cadaveric nerves and fascicles. These measurements revealed that epineurium area, endoneurium area, and perineurium thickness all varied significantly across different donors. This indicates that peripheral nerve anatomy is highly individualized, even at the microstructural level. For example, thicker perineurial boundaries could influence how well electrical signals propagate within a fascicle. These anatomical variations are critical in computational modeling and underscore the need for patient-specific data when planning neuromodulation interventions. Taken together, these findings tell a clear story. Peripheral nerves, especially those within the autonomic system, are far more complex and variable than current models often assume. If we apply the same stimulation parameters to every patient without accounting for structural variations, we risk unintended side effects. Personalized neuromodulation therapies that are grounded in microanatomical data could significantly increase efficacy and reduce risk. Looking ahead, there are several promising directions this research can take. First, modulating the vagus or sympathetic nerves with better structural mapping may improve outcomes for a wide range of chronic conditions, from epilepsy and depression to arthritis and heart failure. Second, these findings encourage us to build more anatomically accurate 3D models for computational simulation. Third, continuing this type of work across more cadavers and expanding to other autonomic nerves can help create a comprehensive nerve atlas. Finally, long-term clinical applications could involve preoperative imaging that adjusts stimulation parameters based on individual nerve structure. This could be a future where neuromodulation therapies are no longer trial and error, but instead tailored like a custom suit, precise, predictive, and personal. And these are my references. I would like to thank Dr. Andrew Schofstall, my parents, and my school teachers who have supported me along this journey. I would also like to thank Case Western and the National Institute of Health for making the human cadaver material and equipment available for data collection. Last but not least, I would like to thank the conference organizers for making this possible. Thank you so much for listening, and this is my contact information here. If you'd like to connect or have any questions about my research, I'd be happy to hear from you. Thank you.